Welcome to Octorum, the literature review for the obscure scientist. My name is Isabel Berthelot, your host, with Oliver Hutchins, my co-host. Stay tuned for more.
Hello, everyone, and welcome to episode 4 of Octorum. If this is your first time listening, welcome in. If you are one of our tens now, potentially tens, probably about like 8 recurring listeners, so happy to have more than 8.
We have way more, Oliver. We've made it big.
How big are we talking here, Izzy?
Actually, we saw a spike again today in listeners, which we published 5 days ago and only really advertised 5 days ago. We have around 60 streams and 20 followers, as in people who are subscribed to our podcast to get notifications every time we upload something new.
It is such a joy. I'm glad we have something here that you deem valuable. You know, I think that's the best sign we've gotten yet, that we're onto something. All right.
Mm-hmm.
Yeah. Well, if you are those listeners who have listened before, welcome back. Welcome in. Yeah. All right. We have a wonderful episode here for y'all today. We're both pretty excited about this one. This is one where just even like the week building up to this episode, we've just been getting more and more ideas, throwing more and more papers into our little communal paper reference. I think it's going to be a pretty good conversation.
We have a very exciting episode that is going to be exploring cloning as a biological technology. There are so many doors that can be open with this conversation. Like Oliver said, we've both been brainstorming this throughout the week, and there's just so many examples, so many different facets of cloning. And I know that cloning, from a science research world, it is something that we use in the lab whenever it comes to cloning DNA or something like that. But whenever it comes to cloning an entire individual, that's not really something that is done unless it's like in a cell culture or bacterial culture. And so from a layman perspective, cloning does seem very sci-fi. But the reality is cloning is something that exists in nature, and it is something that we can harness and use as a technology in ways that might seem unnatural. So there's so much to unpack here. And part of the reason why I'm so excited for this episode is I was even listening to a podcast earlier this week where a woman was saying, why doesn't anyone talk about cloning anymore? Like in the early 2000s, scientists cloned that sheep, famously named Dolly, and created an entire copy of this one individual sheep, and it was the biggest deal, and then we just never talked about it again. And so I was really excited because in my head I knew that was going to be the next topic that we explore in the podcast. But like I said, what many people don't think about often is that cloning occurs in nature very frequently. In plants and bacteria, cloning is something that is very normal. Even in animals, some species have the ability to regenerate themselves. But one thing that I did want to dissect is regeneration truly the same thing as cloning? What is the difference? Because it's not the same at all. I actually read this off of Google. So regeneration refers to the ability of an organism to replace or restore lost or amputated body parts, tissues, or organs. Whereas cloning involves the creation of an identical copy of an organism. So that's why it was such a big deal whenever scientists were able to clone an entirely new sheep, because it was the same genetic individual born at a different time, almost like a genetically identical twin that was born 5 years after the original individual was born. But one question that I had for you, Oliver, is if you could explain cloning using the example of propagating plants from cuttings versus seed, just to give like a completely new overview to our audience about sort of the difference in developing an entirely new individual from one original.
Oh yeah, I could easily explain it from that angle. I mean, that's the best way to understand. It's pretty foundational to most horticultural, botanical practices in the world. So again, you can think of the seed is the baby of a plant. You know, this was a product of sexual reproduction, genetic exchange. This is a new baby individual produced by the mother plant to be able to go off into the world, grow, take in resources, and eventually become an adult. So a seed results in a genetically unique new individual. While if you are propagating something, again, propagating is just the layman's term for it. You can really think about it as casually cloning of, you are taking, say it's your treasured house plant that you got from your mom or a Lowe's, wherever you happen to get this plant. You cut off a stem of it, you put it in a cup of water, it grows roots, you repot it. Now you have two plants. Genetically, they are exactly the same. So, in a way, you can think of them as exactly the same plant, but effectively, they are clones of each other. This is how every apple variety works. Whenever you think about Honeycrisp, like what makes a Honeycrisp apple a Honeycrisp, those are all genetically exactly the same. Those all came from one genetically unique individual that was, I don't know exactly how the Honeycrisp made, whether it was just, intentional selection, hybridization, how genetically involved people were in the process. But once they did produce the Honeycrisp variety, they then take that genetic individual and propagate it and make clones of it until they have enough to be able to produce enough apples to fill the food supply chain.
There is another way that this shows up too in modern culture because, like you were saying with the Honeycrisp, that is one specific branded, trademarked, if you will, type of apple. And like you said, in order to grow it with the same genetic code to have it be a true Honeycrisp, you have to grow it from cuttings because a seed is a combination of two halves from mother and father. It's going to create a completely new genetic individual, to whereas cutting is one in the same from the original. And whenever you're talking about genetically modified fruits and vegetables and plants that are commonly grown in modern agriculture, there are a lot of companies that will patent the genetic code of whatever fruit or vegetable you're growing, develop a name for it, and then in order for farmers to grow it, they need to purchase seeds or cuttings.
What, for whenever like a farmer's trying to grow a trademarked produce?
Yeah, like the biggest example, I was talking about this with Eric the other day because he was listening to a long podcast series about the history of Roundup. And then that caused scientists to create Roundup resistant crops in the 90s that could produce Roundup resistant seeds. Farmers could plant them and then just nuke all of the crops with Roundup because they wouldn't die. And if somehow the company finds out that you're using seeds that you saved or seeds that were purchased from another place, you could be sued. If you were to save the seeds that were produced from the seeds that were given, those new seeds are going to be a different genetic sequence that isn't close to the original. Like you said, if you save a Honeycrisp seed and you plant it, you will not get a Honeycrisp apple. You're going to get something different. All of this to say, nature already clones. Whenever I was researching sort of the cutoff of cloning, like how do we know what species is developed from clones versus what isn't, or even kingdoms? We were talking about how Kingdom Plantae can clone, and that's going to be the topic of the rest of our episode. But then, of course, as humans, we wonder how that's relevant to us. If we could clone a sheep, is it possible to clone an entirely other being? So plants have the ability to create offspring either by sexual reproduction, where you have male gametes that fertilize female gametes, or they can create a genetically identical offspring. Like I said, this is usually rare in vertebrates, but it can happen in fish and fungi. This can happen from spores. And of course, in bacteria, there is an instance where genetic information can be shared, and that's the closest thing that bacteria can get to sexual reproduction. But in general, they pretty much just double their genetic information, separate, and then clone themselves. Whenever I was researching the question of cloning, I went to the regeneration website of the National Institute of Health, the NIH, and I do want to share a quote with you about what I read because this helped me understand a lot better. Because in my mind, I was thinking, if regeneration is sort of a partial cloning of some forms of tissue, like we know that in humans, if you cut your finger or something, it'll regenerate and heal. It'll create a scab, it'll close the wound. But why can't we clone an entire arm and regenerate limbs whenever other species can regenerate limbs, if not an entirely new individual just from one little like portion of it, like what Oliver was talking about with horticulture? And so I want to share this quote. Once again, this is from the regeneration page from the NIH, and it reads, "Some organisms regenerate by first forming a mass of many types of dividing cells, including adult stem cells at an injury. In plants, this mass is a structure called the callus, which grows into new leaves, flowers, stems, or roots. In some animals, such as salamanders, the structure is called a blastema. After an amputation, blastema cells rapidly divide to form the skin, scales, muscle, bone, or cartilage needed for forming a new limb, fin, or tail. Most mammals, including humans, do not form blastemas. Their limited regenerative abilities are the result of adult stem cells that differentiate into the cell types of the tissue or organ in which they were found, which enables the formation of parts needing replacement due to injury or aging. While humans can't regenerate limbs, they can undergo compensatory hypertrophy, which is an increase in an organ size in response to damaged or removed tissue. When parts of the liver is damaged or removed, for example, the remaining portion grows to the original size and allows for the liver to function as it did before. To me, this is relevant because it created sort of this cascade of understanding what could be replicated versus what couldn't be. And clearly, it is very different for different kingdoms, and then within those kingdoms, different species. And so there are some species where they have almost a stem cell type technology that is persistent throughout the species' entire lives. But then in humans, because our bodies are so complicated, we have different levels of stem cell. And so we can't regenerate an entire limb, but we can regenerate things that are more within the range of what that stem cell could do. But then this leads to this entire interesting life of plants, because these are one of the rare kingdoms in which they can regenerate completely from the original. And so I think that this would be a perfect case study to use to understand what is cloning, and then to take apart cloning as a technology in the biology of life.
And then how can clonality inform individuality? There's questions about what is the role of the individual in society. It's just one of those words that if you really sit down and think about it, you're hearing it a lot. And so that makes me say if this is a word that we're hearing all over the place, but not really spending a lot of time with, that's all the more reason to sit with it more. We're aiming here to have a pretty broad conversation that takes individuality in places that you've probably never thought of it before. Yeah, so just the fact that we have established, clonality as this phenomenon, it's something that we are very interested in a scientific research, you can almost say artificial capacity, that is informed by the natural presence in the world already. And again, like Isabel said, the place where we see this really, really prominently is in plants. I believe the number given in one of the papers we're going to cite later on, Duhovnikov and Dodd 2014, which I'm going to refer to as Dodo 2014, Epigenetics, a potential mechanism for clonal plant success. The number that they threw out there is that 40% of all plant species are clonal. So, this is something that we're treating as, people think about, clonality. It's this strange concept, we're doing it in labs, trying to clone sheep. No, this is something that basically half of all plants are already doing at some level in their lives. This is just an inherent natural way about how plants operate in their ecosystem. And so luckily there is a lot of academic conceptual precedent for navigating this. How does clonality exist in plants? How do we measure this? If you think about the collection of data or doing research as a whole, we are trying to look at different individuals, measuring them as different data points, creating effective sample sizes so that we can make experimental claims beyond just what's observed once. We want to be able to systematically and through controlled experimental settings be able to define what an individual is. But if you have a plant that's cloning itself all over the place, you need to be able to say, well, how's this affecting our ability to define an individual or be able to record things on an individual basis?
One inspiration point for this episode for Oliver and I was the research that we were a part of in undergrad during our ecological research fellowship. I did a lot of the mapping for this project and almost the artistic side. That was sort of the strength that I could bring to the table for this as well. This really strange enigma sitting on the top of the Cumberland Plateau in Tennessee, the campus that we were at, which is 14,000 acres. It spans a wide range of the mountains. Our research advisor, Dr. Jonathan Evans, and previous biology departments had known about the existence of this widespread Hillcane, which is in the family of bamboo. It appears almost like sugarcane or short bamboo stalks. Appearing from the ground, these biology departments were tracking its dispersal, seeing where it appeared in different places, seeing if it was still there the next year. And so fast forward to around 2022, whenever I began working on this project, long-term project that had already existed, and we were plotting the movement of this plant over the Cumberland Plateau. And the way that we did this was by excavating the roots. We could see the shoots above the ground. Like I said, almost looks like short bamboo or yellow sugarcane. But then under the ground, it was all connected by this linear rhizome. And it could turn and zigzag and move in different directions, but it was pretty much one main line that was connecting all of these shoots. And so over time, this research was done before I joined this project, but it became realized that what seemed to be a community of Hillcane that they were tracking over time on this mountain was all connected, and it all stemmed from the same mainline root. So what's happening is the rhizome is going all along the mountain, turned out to be one genetic individual. And for most species of bamboo, they produce these yellow flowers. This can also be triggered by fires, so burns can trigger the blooming of these flowers. And flowers are one of the reproductive organs of plants. And so plants use flowers for sexual reproduction. But because this widespread individual, and whenever I say widespread, like Oliver, what do you think the distance was?
When you think like the largest organisms in the world, there's that like quaking aspen in the western United States, a whole hill slope, I think. So, I mean, like, that's the best mental model I really had at this point of just how far one individual would go. Like, when you think of superorganisms like that.
I actually was genuinely asking you, like, I
But
don't know the exact length.
your project was mapping out the distance. I
It
thought
was
you'd be
mapping
like,
the age. It was using the distance to map the age.
oh, okay, yeah,
But
yeah.
I don't remember the exact distance.
Yeah,
And
like
so
spread across the entire plateau was about like 2 or 3 individuals max, right?
we were studying one, but on the entire plateau, we know that there are at least 2 or 3. But essentially, because this was all one ginormous individual, and Hillcane does have the capacity to reproduce sexually using flowers and pollination, we know that that never occurred. Because if this individual would have flowered and produced offspring, then we would have seen genetically different individuals across the mountain, but we did not. It was all one genetic individual that was just ancient, where this mainline root was just cloning and moving along the mountain, and then some areas of the root would die, and they would appear to be individuals, but they really weren't. They were just parts of the main line that had drifted away from the original. But my role in this was using the data from previous years of where this Hillcane was and when. I could plot out the rate and essentially see how much it was moving over time. And if we knew the origin source and we knew the furthest distance that it was now, we could plot a range of how old the species would be. We realized that it was thousands of years old. This one individual was thousands of years old. It was ancient. And so all of this brought about a lot of questions of what is an individual? Because I would have never expected that not just one species, but one individual plant could be thousands of years old, have traveled and created offshoots of itself that is still itself, and now had its own existence on the mountain. And another aspect of this that I want to add before I move on, because this root was a line moving forward and backwards and everything being an offshoot of it, what we could do is sample and sequence the genome of this rhizome in one point and then later down the line in another point, and we could actually see in real time all of the genetic changes that it was taking on from the environment as if it was in itself a timeline. So we could see a line, a linear movement of how the environment was changing the genetic expression of the individual as it aged and moved.
Yeah, I want you to spend more time just talking about what that was like for you personally to experience of seeing this scraggly little bamboo on one side of the mountain and then going to the other side of the mountain and seeing another clump of stems and being like, oh, this is the same plant. Like, how does that even register mentally?
I think it was very surreal realizing that we would have to travel such a distance to even sample and excavate these roots, even though it was the same individual, because it seemed larger than what one individual could do. We even had an ongoing joke of it could have been Jesus Christ himself who planted the original one because it was that old. And so just understanding that this one individual was all over the mountain, spanning very large distances, was extremely surreal and, needless to say, made me feel very small. Although it was powerful enough to span the entire mountain, because we were able to reason that this was one individual, and because it was all one individual, it never flowered ever in its existence. And think about it, this plant was now thousands of years old and never once had it flowered. And I remember my research advisor, Dr. Jonathan Evans, saying the day that it does flower, and it could be any day, it could be any random day, and we'll wake up on the mountain and look around us, and there will be yellow flowers everywhere on the entire mountain because it will all flower on the same day, blew my mind. But one thing that it made me think a lot too, although it was so powerful to have expanded so far and sustained itself for so long, we still felt like somehow it was broken. That was the language that we would use a lot whenever we were talking about it. It was like its clock was broken, and for some reason, this function that it was supposed to do, it wasn't doing. And even, I mean, researchers before me had tried to stimulate the flowering by having controlled burns, by working with different temperatures, and nothing would work. It seemed like it wouldn't flower. But that is another part of what fueled this episode, trying to understand, was it really broken or was it outsmarting the environment?
Mm-hmm. I feel like the other crazy thing about that too is just how that is just so incompatible with what an individual is supposed to be. You know, like individual, just look at a loose breakdown of the Latin roots, there's undividable. Like it should be the baseline smallest unit of what this thing is. But yet at the same time, we're saying, no, this individual exists in all these places at once, and it's thousands of years old, and it's all these different things. It's like, how are those things supposed to coexist? And I think that's the biggest thing I've grappled with or tried to understand. Again, also being in a clonal plant ecology lab, just trying to understand that breakdown of my understanding of individuality. And this may seem like this new discovery, but in terms of plant ecology, this is a decades old concept that just anybody who's worked with plants for more than a few years realizes this is exactly how this works. When we were first discovering this, you know, with the Appalachian Hillcane, I think the full Latin name is Arundinaria appalachiana.
Yes.
In case y'all want to dig into the Wikipedia page there, I'm pretty sure there's a Wikipedia page. But to us, that was a new discovery that was just, you know, scattering our marbles or breaking our brains all over the place. But this is by no means a new discovery in terms of plant ecology. I was going back to try and look at the first emerging understandings of these seemingly paradoxical notions of individuality within plants. I was looking for papers where, all right, when is this like first discovered, coined, explored? I was taken all the way back to a paper in 1974. Kays and Harper, 1974, The Regulation of Plant and Tiller Density in a Grass Sward. This is the first paper to really coin and define the multiple notions of individuality that plants are given to be able to sort of navigate and understand the strangeness of their paradoxical individuality. The whole purpose of their study is they're trying to understand how individual density affects the number of individuals or the population dynamics within a grass population. there was an already emerging plant ecological understanding that, the size of an individual affects how many individuals a landscape could support. If you imagine, you know, a a farmland of just like a farmer's set of fields, and they do the math and they say, all right, we can support 100 cows on all of our pastures here. That's assuming all these cows are normal size. It's like you just got 100 standard cows. But what if you said, well, what if we put mini cows on this pasture? Like cows are just like half the size of a regular cow. If you think about it that way, you could theoretically have 200 cows on the same amount of land supported by the same amount of resources. The concept there is to understand that there is this trade-off of like the more one individual needs, the less a given environment can support the number of individuals. And so, Case and Harper, they were looking at plant ecology literature. There was that emerging understanding. People were doing the mathematical equations to understand how many individuals could be supported in a landscape. But they're saying, oh, we've been looking at this, assuming you have a standard size individual, they're reproducing more. There's almost this kind of like maximum to how big one individual could get. But they're saying, oh, that doesn't really apply for clonal plants. Because if you think about it, you could have this one genetic individual just getting larger and larger and larger over time, almost competing with everything else around it. But they were coming in as clonal plant ecologists saying, well, hold on, the fact that plants clone themselves completely change how we have to think about how individuals within a population are competing or living with each other. So they are trying to navigate how is that affecting reproductive capacity in the population? Are they all just shooting each other in the foot, making it so it's harder for everyone to have kids? Or is this not even about, producing new individuals, reproduction, offspring? What is this even really about? That's how I came to understand the conversation they were having that they expressed in the form of a paper. And because they were working with clonal grass species, they were having trouble just understanding it from just like a purely genetic individual angle. Plants are highly clonal. Anybody who's worked in horticulture or whenever you are just taking cuttings of plants, like if you just have like a house plant where you have like a little pothos vining off or producing stems, you cut it off, repot it, you're working with clonal plants. That's just an inherent factor of how they've been going or how they live that's been understood well before academics tackled the question. So they already went in knowing that plants are clonal, affecting how we understand how these populations regulate themselves.
And I think that it also really sparks the question of why do some plants and other organisms who also have the capability to reproduce sexually, why do they choose asexual reproduction, which is also referred to as cloning?
Yeah, that really is the big question here because most ecologists treat it as like the word that comes up a lot is broken, saying like, oh, there's something broken going on here, either saying these whole species are broken or saying something broke that made them behave this way. Because again, if you look at evolutionary biology, Darwinian evolution, all that theory says is that it is all about sexual reproduction, rearranging and selecting genes over years to produce better and better species. So you'd think that like the most evolutionarily fit or like the best species as a whole is just constant reproduction, constant genetic reshuffling, just to increase the chances of producing higher quality genes. To have a species that's instead of creating new generations saying, nah, I as one individual am going to live longer, that almost feels counterintuitive to evolution. So a lot of clonal plant ecologists have tried to understand why they do this because again it seems counterintuitive or against the whole purpose of evolution. This is explored a lot in The Loss of Sex in Clonal Plants, a paper by Eckert 2001, which is kind of like a broad review paper just looking at various hypotheses around understanding what may be leading plants to clone themselves and understanding the consequences, whether it's beneficial, whether it's antagonistic to species survival, and also trying to understand practically, logistically, how is clonality even happening. There might be times where, times are tough. It makes more sense to stick it out, live for a couple more decades instead of reproducing more individuals. But even still, it's coming across that same angle of this feels like this species is refusing to evolve. This feels like it shouldn't be the best strategy.
I don't know if I would fully agree with that. I think the paper, The Loss of Sex in Clonal Plants, that Oliver's referencing from Christopher G. Eckert, Well, firstly, he's setting the scene by reminding us of these pillar statements. Plants can reproduce sexually or asexually. They can clone or they can reproduce sexually from male and female parts. But part of the argument that he's pitching here is because here he's talking about what is the development of having this clonal regeneration in general? Where did this come from? Why did they do it? And part of what he's talking about is how these environmental factors that these plants reside in heavily influence either forced sterilization, as in there aren't mates in the area to be able to reproduce sexually, which means that in order for the species to survive, then it has to lean more towards clonal reproduction. Or if the environment has factors like temperature or other physical barriers that would prevent reproduction, then this would also allow for another potential way to have the species continue on. You understand the perspective that I'm pitching?
Oh, absolutely. No, you're coming with a much better understanding of the paper than I did. If I could frame how I was coming across it better was again looking at more of like a persistence model of if sexual reproduction isn't an option right now, it makes more sense to persist in this environment instead of it to go extinct within this one space where sex wasn't able to happen. But then he also explores how there could be consequences to that. Like again, the one of the big genetic concerns there is sterility. The constant cloning of an individual, constant cell division. The understanding is that there are genetic mistakes that accumulate over time. Through sexual reproduction, the idea is that a lot of those mistakes can be caught, corrected for whenever just like genetic recombination can happen. But if you just have this individual that just, like copy and pasting, copy and pasting, copy and pasting, or like imagine if you had a document that instead of just like saving it as a file and reprinting it from the PDF every time you wanted to use it, you would scan it and then print out a new copy. Every time you'd get a little smudge on the paper or like spill a little bit of coffee on it or like the paper gets wrinkled a little bit, that will be picked up by the scanner. And then every time it's reprinted, that'll be there. So you can imagine, after 10, 20, 30, 100 times of rescanning and reprinting this document, it'll be a mess after all those reprintings. That's pretty much the same fear of plants that clone themselves too much, of if these plants keep going for too long without reproducing sexually, so many mutations will accumulate where this plant will no longer even be able to sexually reproduce with other individuals and it'll have no choice but to keep cloning itself. And again, that's where that language of broken comes in. You have these individuals that are almost like sexually isolated, no longer able to interact with their species. They're just going to keep on living until something kills them.
Right, and I will say I do remember I talked about the Hillcane enigma at the beginning with Arundinaria appalachiana, which did not flower, never reproduced sexually, but yet had this very long continuation of its life and dispersal for thousands of years over very long distances. And I remember during that time, we were thinking either this plant is completely messed up or it's the smartest organism that's ever lived. And so, something like that, it does really challenge this idea of what is success? What is biological success? Is that smart or is that broken? And so it does bring up this whole other question of what is the underlying intelligence of life here. But one quote that I wanted to share that I think really well summarized the conclusion that Oliver was talking about. In this paper from Eckhart, he was talking about how the origin of clonality in plants comes from environmental factors that force it to reproduce asexually. But like Oliver was saying, later down the line, that might lead to future forced asexual reproduction because now the plant doesn't have another choice. It can't sustain sexual reproduction. So here's one quote from page 288, and it reads, "The prevailing hypothesis for the evolution of genetic infertility is that sex is lost through the accumulation of sterility mutations in populations where sexual recruitment is impaired by the environment." So that just really well summarizes what we were talking about. Although Eckhart does bring up these questions of, is this genetically unfavorable to have constant cloning rather than the genetic diversity that sexual reproduction brings. He also calls for more research around the topic. He calls for more research around how reproducing clonally would affect the ability to reproduce sexually later down the line. He does make it clear that there isn't a lot of information around the topic, which I think is a beautiful segue into another paper that we found to help support this conversation. You have your fun way of saying it.
What, Dodo 2014?
Yeah, because I think that this paper that we're about to introduce almost challenges this idea, because it argues that clonality is the smartest evolutionary possibility.
Mm-hmm. Yeah, and I see what you're saying there too, with just kind of like the Hillcane linkage about like either this thing is broken and lost, or it's so much smarter than we are.
Right, because is it
Mm-hmm.
losing? It's lived longer than we have and it's spanned longer a distance, so what if it's not producing new genetic individuals? It's still surviving.
Yeah. Yeah, that feels so much more right to me. Like, who are we to look at this, this population where amazing bamboo that like owns this mountain and say you're broken? Who are we to say that? Like it feels like we're missing something in saying that. Moving in that direction of seeing how this might actually be the smartest thing we've ever seen, which brings us to Dohovnikov and Dodd 2014, which is a mouthful, so I'm just going to refer to it as Dodo 2014. Dodo 2014, Epigenetics, a potential mechanism for clonal plant success. They're borderline proposing how there might be an alternative to genetic exchange through evolution via this combination of both clonality and epigenetics. Yeah, so I have a direct quote from the paper. Clonal plants may be one of the best examples of organisms taking advantage of epigenetically derived acclimation as an alternative to the slower mechanisms of adaptation through natural selection.
I highlighted the exact same quote.
Yeah. I mean,
Amazing.
it's hard to
It
miss that.
does, yeah, it does perfectly summarize what they're trying to say. And I think now, because our viewers have been built with the education that we've given for the past 3 episodes, now that we understand the role of epigenetics, which once again is the way that the environment changes the expression of your DNA, right? Because even though all of our cells have the exact same DNA, and even though throughout our entire lives we have the same DNA for one individual, the environment that we're in is going to cause silencing and enhancing of some of those DNA segments to change our expression, which, although it isn't deletion and addition of DNA information, it's essentially having the same effect. And so, what is being proposed here is if you mix the replication of one genetic individual over and over and over again and allow that genetic sequence to live in different environments and different times with different needs, epigenetics, which is the role of the environment on DNA expression, will still allow for some form of evolution. It's almost like, I mean, whenever I read this, Oliver and I, we were both amazed because this almost does seem like a shortcut to waiting for natural selection to occur with sexual reproduction. Like, think about it. If you're cloning the same individual over and over again and then putting it in a bunch of different scenarios, but the environment can still affect how DNA is expressed, then you're basically allowing for testers of one individual in different environments. And that one individual, even though the entire genetic sequence is the same, it's still going to pass on those epigenetic enhancements. And so what's really interesting is think about Eckhart, the loss of sex in clonal plants. Part of what they were proposing is this fear that clonal reproduction actually decreases genetic diversity in communities and causes forced sterility later down the line. But what this paper is proposing is there actually isn't that much difference in genetic diversity in communities that replicate clonally versus non-clonally, which is insane to think about because it should be one identical individual after another. It should be completely the same, have zero variation whatsoever, but they're actually arguing no, that isn't the case at all.
One of the things that reads as funny to me is in that same quote, they describe epigenetically derived acclimation as an alternative to the slower mechanisms, like almost dissing natural selection a little bit, saying like calling it the slower mechanisms. But I mean, there is some truth in that. Again, like any evolutionary biologist would say, like the formation of a species, like natural selection happens over the course of like millions of years. Like even if what we're looking at here with like clonal plants plus epigenetic accumulation, even if that's happening for an individual over the course of centuries, that's still compared to millions of years insanely fast.
If you're thinking about the genetic variation that would come from sexual reproduction, which we're familiar with, that would be over generations. To whereas the genetic diversity that epigenetics allows exists in one lifetime. And so it is really enhancing and sort of speeding up this process, or at least proposing a theory that it could do so in a way that's really interesting. And one thing that I wanted to note, whenever we're talking about the ability for the environment to change the individual and how clonality could be a technology to help adaptation, one of the quotes from this Dodo 24 paper is, open quote, broadly speaking, epigenetics is a form of plasticity, end quote. In order to be persistent in an environment, what needs to happen is the capability to evolve around whatever changes is occurring in that environment. And I really like the word of plasticity here and representing that sort of visual of how evolution is conferring. I also want to share a quote later down the line. This is on page 229. And it reads, modularity is an important factor in the success of plants, and clonality is the extreme example of modularity. At the beginning of this episode, we were talking about the technology of clonality whenever the environment does not allow for mating or sexual reproduction, whether that be because of separation of male and female populations, which happens all the time in the plant world. But whenever there's an environment where sexual reproduction is not available, clonality is the technology that's used to allow for reproduction. And then here, now clonality is used again as a technology of rapid sampling of the environment to see which genetic composition would be better fit for the environment. And then I think from here, earlier, I talked about the Hillcane enigma, and we're going to reference that all episode. But one thing that I emphasized, and I'm not sure if I explained it well because it was so profound, the fact that this long rhizome, this linear line, could almost be used as a real living timeline because you could sample the root, you could sequence the genome of the root at the origin spot, and then you can sequence the genome of the root 200 meters away and see the differences, see which areas of the DNA was muted, silenced, or enhanced. You can see those epigenetic effects, and in that way, it really is a beautiful form of being able to monitor these modulations that paper Dodo 24 is talking about.
Yeah, that's a much better segue for that. Yeah. So the whole notion of clonality in plants is not a new one. It's new to us, but they have understood this or have been studying this for decades. So if we want to use the language to understand these clonal and genetic identities in plants, we can go back to Kays and Harper 1974, the regulation of plant and tiller density in a grass sward. This is the paper in which these crucial terms that are used to understand these concepts of individuality and clonality were coined. They use the terms genet and rammet. Genet can be commonly translated to the genetic individual, while rammet is more like the physical individual. So the genet is the individual that's been united genetically. So, like, again, we talked before about how a seed is a genetically unique individual, while a cutting or a propagation is a genetically similar individual to where it came from. So if you want to think about what all falls under the concept of a genet, it's everything that comes from that seed. So even if that seed germinates, roots, turns into an adult plant, and one day that plant clones itself and, sends off an offshoot with its own roots, own stem, own leaves, and physically it no longer has anything to do with the plant it came from. Even though they are physically separate, genetically they are still identical. So they're considered part of the same genus. So they're still the same genetic individual, even though they're different ramets. They are physically independent. They are no longer connected to each other. They are operating independently. They are, in that way, their own individuals. So for understanding clonal plants, you kind of have to constantly be going back and forth between these dual modes of individuality, where you're thinking about the ways in which they're genetically united. At the same time, you're thinking about all the ways in which they're physically independent.
Going back to the paper, The Regulation of Plant and Tiller Density in a Grass Sward, I want to share this one quote, which honestly, for the entire podcast, for me, this was like the most powerful quote out of all of these pieces of literature. And I think that it really brings home the point that Oliver was making about using this understanding of a very vast genus versus one unit of a ramet, and how that really impacts our understanding of individuality. I want to read this quote. Open quote. A higher plant is, in a sense, itself a population. A classical interpretation of the shoot system is of a population within which a leaf with its axillary bud constitutes the unit. The whole plant is thus a matriarchal tribe of shoots, many generations of individuals living together, independent upon one another. End quote. To me, that was a very powerful visual. I want to bring back what I was saying in the beginning about how we understand and comprehend what clonality looks like for us humans. Like I was saying, it sounds like such a sci-fi concept, but in reality, it's something that exists in nature very frequently. But us as humans, we cannot comprehend what that would feel like. There is a technology to create an entirely different individual than oneself. But what we cannot comprehend was what I just read, in which on the trunk of a tree exists entire communities of different individuals, and in that way, one tree is an entire village. Imagine if you as an adult, this is going to be a crazy way to explain this, but imagine if you as an adult, I'm like 23 years old. Imagine if I grew an entirely new limb whenever I was 23 years old. It was different enough from me, Isabel Berthelot, and my mind, where they had separate consciousnesses. We can't imagine what that feels like to carry a community, like of even ramets, what we're talking about. We can't comprehend what that means for us as humans. But in that way, it's beautiful that there is one individual in nature that exists and lives that way, in a way that is so different from our existence as humans. It really does challenge the concept of what is an individual.
And I think the other really amazing thing to add here is that Kayes and Harper, they were not thinking about epigenetics at all when they were having this conversation, looking at higher plants as a population in of themselves, even though they are genetically the same. But if you think about each one of those ramets as developing its own genetic identity over the course of its own life, you really do have this emerging sense of an individual forming a genetically diverse population just based off of the diversity of experience that it has lived through. And I don't even know how to, like you said, there's like not really any good way we have to think about that notion of individuality.
How do you know if you're part of this one individual, if you're a completely separate entity that exists within that one genetic sequence that has many different clones within it? You wouldn't know. And so it really does make me challenge sort of what my role is, both as an individual in myself, but as an individual thinking about what if my community is an individual. That was a crazy way to say it. I'm not sure if that makes any sense. But do you understand? Like, is there a way that you could rephrase that better?
I think what I'm hearing is, even though it feels very incompatible, it's still making you wonder because it opens up the concept of an individual so broadly, it can open up my own understanding of myself as an individual. That's kind of where I heard you coming from. Like, even if you didn't quite know how to go about it. With that, I'd like to start us from a pretty broad place with just a question of what is the role of the individual? I think culturally, we have a lot of points in which we can tackle that conversation. The whole idea of individualism is pretty big in terms of like a cultural conversation. You have individualism as a Western cultural prominent trait, a pro and a con. Like some people say we're too individualistic, some people say we aren't individualistic enough. Going back to a paper from episode 2, Torday 2021's Cellular Evolution as the Flow of Energy. To those who listened to episode 2, you will remember that this was quite a bold paper reassessing how evolution could be understood as the flow of energy more than the flow of genetics or reproductive goals or anything like that. So Torday was going in saying, is evolution really about making species more and more genetically refined for whatever purpose? Or is there something else going on here through which we can better understand the purpose and meaning of evolution? I bring this up, one, because I think it's good for people who aren't as in depth or like as involved in academia to know that you can go back to a paper as many times as you want. Rarely are you looking at a paper once and just saying, I'm done with this, there's nothing to say. Most of the times, whenever I'm citing a paper, it's because there's one sentence in there where I'm like, that sentence is good. That one sentence helps me right now. So you can not only continue citing the same paper over and over again for that same reason, but especially with like huge papers with bold claims like this one, you might be finding more and more reasons to revisit it in the past. And this is one of those cases where, before we were just looking at, because we thought just the concept of energy as a way of measuring evolution was phenomenal. I bring this up again because I think it's just good to bring us back into that playful state of asking, looking at preconceived notions of what evolution is, generation after generation, making a species better. The role of a species and your role as an organism, as an individual in the species, is just to be a part of this stage where genes are selected for in a survival of the fittest kind of way. Is my life just about, seeing how I fare in the world and hoping my genes mean something? There's just something that feels very deficient about that, but I never really liked that answer. So I feel like this is a good place to go back to say, how could we potentially break out of that? Or like, where are the places where it's not fitting into what feels right to us?
Especially in episode 2, whenever we were referencing Torday's paper, we talked a lot about the role of culture and the role of community and using community as the technology.
Another reason I'm revisiting Torday is even his new model of the purpose of life. It still treats the individual almost as an intermediary. We're almost still the middleman of life, but instead of, exchanging or changing or refining genes, we're just trading energy to some greater purpose. Maybe we're just vessels for the Big Bang still enacting itself within us, which is cool and beautiful, but I still don't like how it almost comes off as, again, kind of reductionist towards the individual itself. And that left me wondering, is there a way we could actually take the individual and elevate it, and instead of just treating it as this intermediary stepping stone towards something better? It's like, evolutionarily, the individual is just a vessel for genetic or energetic exchange. Socially, the individual is just a cog in the machine of the economy. I'm wondering if there are ways where we can build upon it, expand the concept of an individual, and make it more than just a piece of the pie.
Yeah, and I think in the last episodes that we've had, like all 3 of them, we've discussed the role of an individual in the existence of things that are physical science, not even biological science. We were talking about entropy, we were talking about energy, and then we talked about the role of an individual in a community. And I feel like now we're talking about what is the role of an individual in the scope of genetics. And so I feel like that's sort of what this episode is about: individuality on a genetic level and what the significance of being a genetic individual is. And then how does this impact us? How does this relate to us as humans?
I think there is something very resonant about looking at individuality this way, not to be restricted by like physical, genetic bodily identity as much as spiritual or emotional identity. That sense of plurality, complexity, the idea that we cannot just be defined into like one single understanding of what a single person is. I think, I mean, for me at least, I feel like clonal plant ecology has really spoken to that. I mean, one of the very real places I felt that I think has been in grief of just that sense of, after my papa died, just trying to understand that experience and having that very strong sense of, I'm still here, I'm still living in what you can call my entirety. But at the same time, there's this sense that, like, not even like a version, but like something equally as whole is no longer here. And I think as I was trying to make sense of that, I was working with, basically my version of the Hillcane, Dicrostachys cinerea, sicklebush, a shrub native to the South African savanna that I was doing my honors research on. This shrub that was understood to be a clonal plant, so as we know now, creates new individuals of itself that are self-sufficient, just cloning itself in the landscape as a way to survive. And the whole project was trying to understand the role of clonality, of when is this plant cloning itself, when isn't it, and understanding it explicitly through the lens of fire and herbivory. About saying, all right, this plant exists in the savanna. Every few years, it's just burned down to the ground, and constantly it has herbivores coming in, eating away at it. So I spent that whole summer just looking at this plant that, even though like the genus was facing this threat of just being burned and eaten, it was constantly able to create new ramets, new physical individuals off the sides of itself in response. So at the same time as I was grieving, I saw this plant that was truly being eaten alive and creating new versions of itself in response to it. And that felt very true to the process of grief to me, of the sense of, even if there is this own individual that is me in its own very real way, that is facing its threat, that may no longer be here, that may be dying away, there is still this whole other equally true, equally whole individual that is existing outside of it. And I think that's the best way I've tried to be able to make sense of it, or like the ways in which, without even trying, this insane notion of plant individuality has applied itself into my own life.
I just sort of had this really deep understanding with cloning, you're not changing the individual. You're just being more persistent with that one individual and trying over and over and over again. And there might be one environment where it really does work. There might be one environment where it is optimal and you're like, okay, great, this is what it was all for. But I do really appreciate that level of persistence of having the same thing over and over again and just trying again with these different environmental factors, trying again with these different simulations. I do really think that in that way, like I did just realize it's almost the inverse of natural selection. It's not letting nature do any of the deciding. It's not letting nature change you in any way. It's just trying again, over and over. And I
Yeah,
think that, yeah,
it's almost not even like saying I'm not giving up.
there is this largeness that comes with that persistence of knowing even if you fail in one environment, it doesn't mean that you failed altogether. It just means that you failed in that one environment.
Mm.
But that same you that exists in the same capacity, like what you were talking about before, can thrive in another environment. Just because one thing fails doesn't mean that everything's going to fail. It might mean that 5% of things are going to fail.
Mm-hmm.
It requires that persistence. And then, of course, epigenetics is a good mirror for like trying to understand the ways in which we could let our environment guide us, but not change us, but guide us. And so I think in that way, it's another aspect of persistence as well.
Mm. The more ways we talk about this, the more ways in which it feels resonant. I mean, like, there's also that whole sense of just, who are you as you age? Like, were you the same person this whole time? Can you think of yourself as like the same person you were a decade ago?
Mm.
Because at some level, you have to say, even now, I feel like just, 24, so I can't even act like, I've weathered the years and have reached this wise state. But I feel like even I look at myself a year ago and my most natural response is saying, oh, that was a different human being. Like, that was a different iteration. Like I feel like the word like
Yeah.
iteration is one that comes up a lot in thinking about personal growth of saying like I have to recognize that that was like a whole, person I was and being, but at the same time it feels other from who I am right now. S-
Mhm.
So maybe if we use the language of the gene, maybe under the spiritual gene or just the idea of like this individuality that contains all other individualities, there is that unity of saying, oh, that is all me. But at the same time, there is a completely separate me that I am right now that has nothing to do, that is in no way connected to who that was.
Mhm. Interesting. Just because something is different doesn't mean that it's not me. There could just be many me's. Like it seems like that was sort of what we just talked about, and
Mm-hmm.
I do want to bring it back to, because it's interesting, like even though it's not directly what we've been discussing, I do want to bring it back to cloning in the human world because it is an interesting thing for us to think about. As we know, sheep have been cloned, some vertebrates have been cloned. I do want to provide a very brief explanation for the technology that was used there, just for people who were interested. And you're like, well, you're a science podcast. Why don't you talk about it? Let me take a couple steps back. Whenever I was talking earlier about regeneration versus cloning, I gave the example of like, okay, well, us as humans, we could regenerate skin. We could sometimes regenerate liver. If you have surgery on your liver, you can regenerate it. But we're not able to regenerate everything. Some animals are able to regenerate limbs, but humans obviously cannot. And part of that is because of the specificity of certain stem cells. There are so many different types of stem cells in your body, some a lot more specific than others. And so, in order to regenerate a very large unit, the largest being the entire individual, you would need to have the stem cell of all stem cells, which is the embryo, right? Like, that is the embryo. But like plants, whenever I read that quote from the NIH, the regeneration page, and it talked about the structure called the callus in plants. That's why plants are able to regenerate every aspect of itself, the roots, the shoot, the leaves, the flowers, whatever, from one segment, because they don't have as much specificity as what animals would have, especially humans. But the way that scientists were able to clone whole animals was by basically engineering that embryo. And so the way that that was done, this was interesting because it's not just introduction of DNA, it's introduction of an entire nucleus. And so essentially what happens is an unfertilized egg is taken from a mother, and then the nucleus is essentially replaced with the nucleus of whatever individual is being replicated, and then from there the embryo is stimulated to grow and then implanted to where it can develop. And so whenever it gets to that like organism stage, multicellular fetal stage, it has the same genetic composition as what was being replicated. I did just want to add that because it is interesting, and I'm sure if we're talking about cloning of animals, like people want to know how that happened and what the technology was. I do think that in theory it's possible to clone a human. I would need to pull up literature to fully support that, but in theory, it would seem if you use the same approach, it would be possible. It's just obviously a lot more ethically concerning. And I didn't read it, but there are, like, I'm pretty sure it was Bush. I'm pretty sure it was President Bush. I'm not 100% sure, but there was sort of a train of research that was done, not just in like the capability of cloning humans, but also the ethical considerations of it and the potential dangers of it, basically. Oliver, how would you feel if you were cloned?
Like if there's just another
Would
Oliver.
you feel like as I don't know if you ever watched the show. Oh shit, what was that show called? It was so good. Like with the innies and outies. Severance.
Severance, yeah.
Yeah. To me it's almost like that. Would you who do you think would be more important, you or your clone? Would you feel like the original and your clone just, an invader? Or would you have some sort of connection?
Oh, Severance is such an amazing tie. You're right, such a crazy direction to bring it in. Oh, I do not. I don't even know how. I feel like I would just like break down crying, like because wouldn't even know what to do. I don't know.
Because I
Which
think
one would
I
I
would have,
value
like,
more?
even though I wouldn't want to, I think I would have some negative feelings.
Yeah, like you wouldn't want to affirm that that was like. It's like, oh, surely they're less than me somehow because it
I'd be
does
like,
threaten
no,
your.
I'm the real me.
Yeah, yeah. Well, no, I feel like that's a classic cartoon plot. Like there's like a doppelganger or evil twin, where it's like, no, I'm the real me. No, I'm the real me. No, I love that. I don't know. I feel like I would. I feel like my response would be surprisingly lackluster. Like I feel like there's already enough stupid stuff going on that I feel like if I just saw a clone of myself, I'd just be like, whatever. Could you do the laundry while I do the dishes? And like I feel like we'd make it work somehow. I don't know, I'm gonna turn that on you. Like, what would you? Oh, wait, I mean, I guess you already said a little bit, but elaborate on what you would do if you did have a clone of yourself.
I guess using the conversation that we just had, my mind is different than where I would just be normally if you would have asked me this yesterday. But using the conversation that we just had, I would want to see what I would be if my environment and my life choices were completely different.
Mm.
Like, what if I was like a lawyer? Which, look, my brother's a lawyer. I appreciate all lawyers. I would never be a lawyer. Like, ever would I ever go to law school. And so I think just watching would I succeed or would I fail, like the same sort of evolutionary outcome, like what environment works? Is the environment that I'm in now the best one? I don't know. Like, am I
Hmm.
doing the best thing for myself as an individual to succeed? I don't know. It would be interesting to watch another trial run where all the choices are different.
Yeah, I mean, I'm imagining even more insane hypotheticals here. What if I had like 30 different little cloned Olivers. And when they were kids, I just say like, all right, you go in that direction, you go in that direction, you go in that direction. Just sending them off to their own peril. See how all of them do in their own circumstances. How could you imagine all that playing out? These 11 did horrible, but that one survived. So, hopefully whatever he figured out over the course of his life will be inherited in whatever kids he has from here. Or whether he decides to clone himself, it doesn't. This is just getting hard to follow. No, it's a lot to think about. I think I'm pretty happy with just Oliver number one. Like I don't think I think I'd rather just stick with the just one Ramit. Like I feel like that's a good way to
Yeah.
do it. It almost kind of like validates what happens over the course of your life, and I think that's what I think I was trying to get at before at the very beginning of this whole idea that there's this genetic reductionist view of you're born, a whole bunch of stupid stuff happens, you have kids, then you die, and then they repeat the cycle. But with this clonal epigenetic strategy of existence, to me, the two things it affirms is that there's a reason for you to stay here. Like there's a reason for you to experience and make it through tough circumstances, and that there is something you give to the well-being of your species by doing so. Your goal isn't to just, have kids and give up. It's to experience as much of the world as you can so that you can, in of yourself as well, create something better to give to the people around you.
What is being passed down and the vessel that you are for is important. It is development, it's continuation, it's moving forward. That's what you are, that's what your life is.
To me, all of this literature exists to affirm and support that intuitive truth in you. This is science saying you were right all along. Your life is more than this. I think that's the best thing it can give you.
Thank you so much for listening. More importantly, thank you for supporting the Octorm project. This is bigger than just learning about clonality and evolution. It's about strengthening the muscles in our minds that allow us to maintain our curiosity and come up with new ideas in a world that is full of technology that allows you to not think. In the show notes, as always, I will cite the papers that we referenced. I will include the NIH Regeneration webpage, and I will also include the webpage of Dr. Jonathan Evans and the Hillcane research that we spoke about. Thank you so much.